Delighted to have Suzanne Anker here, chair of the Fine Arts Department at the School of Visual Arts. One of the first art schools anywhere to have opened a bio-art lab in the last few weeks. So we have artists becoming scientists, growing things, and who knows what. And so, here she is.
Thank you so much. Thank you so much for inviting me. Can you hear me? Yes. Can you hear me now? Yes. Okay. All right. Well, now for something completely different. I'm going to be talking to you about this interrelationship of art and science, which I can say has been very antagonistic, contentious. It reeks with envy. It is sort of the kind of dialectic between a kind of freedom and rule. And I think that what we are seeing now in the hybrid practices that are involved in both art and science are something that one can take quite seriously. The eminent art historian Leo Steinberg, in his essay, wrote about this issue. And he says in this essay, art and science, do they really need to be yoked? And he exemplifies the gifts of Leonardo, which of course is the prime figure in this, as producing cultural offerings which, unlike his scientific work, which is surpassed, is a creation that is about the life of the man. And the question of what do we do with works of art or texts, etc., that really don't operate in terms of measurement or repetition or observation alone. Can we ask the question whether these kinds of intersubjective works add to the lexicon of knowledge and the human condition? And I think that the answer here is yes. There have been a lot of discourses over the last several decades talking about the in-betweenness of categories, and that has circulated within the corridors of the visual and critical studies departments and the science studies departments and the art historical departments in universities. Artists are now engaged in creative research in scientific laboratories, while all science institutions employ highly skilled design teams to create visually compelling images. Does that mean that on the cover of Nature or Science magazine, that the best image is the best science? So in a way, the relationship of image making to science has become a way to gather attention. The arts have also played a significant role in helping to communicate science to a more general public. And I would say that there are questions posed by David, and a very hard question, is what can artists do to inform science? And before I cite certain theoretical and historical examples, I just want to raise some comments about this morning's presentations. And if we are going to take this conversation further, then it's very important that we differentiate our terms. We can't say that the aesthetic is all about beauty. How do we term the anti-aesthetic in terms of science and art? What are the social consequences of beauty as a cultural marker? We've seen that kind of play out in the social sphere with Stalin and Hitler. What discourses do these beautiful scientific images refer to? Are they just artifacts of material processes? So these are some of the questions that I think need to be parsed to fully understand what kind of dialogue can eventually take place between artists and scientists. The first concept I want to talk about is a way that art has influenced science. As early as Homer's Iliad, the term chimera comes into play as an imaginary creature, a fire-breathing she-monster, having a lion's head, a goat's body, a serpent's tail, and it was a monster of incongruous parts. Another definition of the chimera is a fabrication of the mind, an unrealizable dream. But more recently, it has attained a new meaning in the dictionary, which is an individual organ or part consisting of tissues of diverse genetic constitution. So what starts out as a literary term or as an imaginary fable ends up as a real entity in the scientific laboratory. You can see the chimera in the red figure painting on the left, whereas the chimera plush, which was designed in 2005, is a popular culture variation of that. In 1986, Dr. David Au and his team combined the genetic sequences between a firefly and a tobacco plant. And when I had seen this, and I was sort of starting to get involved in the intersection of art and genetics. And when I had seen this, I actually gave him a call to find out why he was doing this. I asked him if he was making cigarettes that glow in the dark, and he said nothing of the kind. He was kind of using these as model organisms to eventually be used to see how diseases sort of affect different animals. But what we are also seeing in this technique is another technique that is derived from 20th century modern art and later picked up by the surrealists as well, which is the cut and paste technique of collage. And what collage does is it is not just a visual technique, it is not just a way to create pretty pictures, but what it does is it changes the ontological status of the entities involved. So for example, if you have entity A, which is a firefly, and you have entity B, which is a tobacco plant, what you get is entity C. And what does that mean? What kind of formulation, according to Linnaeus's discrete boundary, are we talking about? And the same thing is true in Max Ernst's image here, which is gouache, ink, and pencil on printed paper. Max Ernst was very interested in science magazines. In fact, this piece is a cut-up version from La Nature. Most of you are very familiar with Alexander Fleming, and he was quite a strange character, I must say. And he's known as the father of penicillin, and he was also known to keep a very messy laboratory. Okay? And as the story goes, he had left for a little vacation, and he had left a petri dish containing the Staphylococcus culture open. And when he returned to the lab, he found a circle of fungus growing in his abandoned cultures, which had killed off the bacterial cultures. Hence, by this unorthodox methodology, or chance, penicillin was garnered as well as other discoveries. This was not his only discovery. But his ability to recognize these occurrences was extremely profound. Many of you don't know, I assume, that he was also an amateur painter. He was painting at the Chelsea's Art Club in London, and then in his spare time in his lab, he began painting with bacteria. And this is what Rob Dunn from the Smithsonian talks about. These are some of his works of art, Fleming's works. He said that Fleming would fill a Petri dish with agar and then use a wire lab tool called a loop to inoculate sections of the plate with different species of bacteria. Each bacteria would produce a different color pigment. The paintings were technically very difficult since he had to find microbes with differing pigments, and then he had to time these inoculations so that the different aspects all matured at the same time. Pretty amazing undertaking. Another messy studio. Francis Bacon's studio, which is now on permanent view in Dublin, his home of his birth. And this was essentially a move of his entire studio, all the debris and the trash from his studio, which included a tremendous amount of images from medical imagery, forensic pathology, assassinations, shattered limbs, corduroy, and anything else you can think of. Paints, cups, slash paintings, oil paints, etc. You can see this for yourself next time you are in Dublin, because it is on view permanently. You can't go in it, but there are 3 points of visuality in which you can look in it. And the question is, why would anyone want to move a messy studio like this and put it in a museum for future reference? Well, one explanation could be that it may be the databank of Bacon's creative energies, same way that Abby Warburg, the eminent art historian, collected images from disparate sources to put them in a kind of memory atlas. We see that again today in the work of Gerhard Richter. So this is something to consider about the kinds of happenstance that the creative act is involved with. Muybridge was very interested in Francis Bacon was very interested in the movement studies of Edward Muybridge, who was a photographer. And he had many of Muybridge prints all over his studios. Of course, you could see they're rather unkempt, and that was a way for him to add new meaning to the work. And this is what Bacon says. He says, "My photographs are very damaged by people walking over them and crumbling them and everything else. And this does add another implication to the image of a Rembrandt, for instance, which is not Rembrandt's. Among his collection of images, as I said, were these diverse kinds of combinatory practices. The photograph of Bacon's former lover, George Dyer, on the right, although it is photographically based, you can kind of see it seeping through as the prototype for a Bacon painting. Edward Muybridge invented the stop-motion camera, and which allowed him to visualize movement in its unfolding successive phases. He set up an array of stop-motion cameras, and he photographed the subject moving through space. He photographed animals and men and women, and even photographed the disabled, which I'll get to in a minute. Animal locomotion captures the movement of a horse, which until this time had been thought to essentially have its legs spread in a different way. But he had proved here in this moment of photography that the horse is shown that all 4 legs leave the ground. And his photographic work documented this fact, which was contrary to the depiction at that time. This was a project he worked on with his patron, Stanford, who later published a book of Muybridge photographs and research as his own. And the funding for Edward Muybridge was taken away by the Royal Society since Edward Muybridge did not have enough scientific credentials. Back to Bacon and Muybridge. Muybridge was also the first one that was able to capture the kinds of pathological gaits that are evident in polio victims or people who are having seizures or other kinds of neuronal disturbances. And for that, I think, again, that his images of these, of documenting these were explicitly studied by scientists for their medical value. The piece on the right is the diagonal piece, which from 1901, when this was shown, it caused an uproar because it sort of made things look like this was done in very poor taste and perhaps didn't show enough pathos or perhaps needed to be in the private inner sanctum of scientists. But Francis Bacon took a look at this and did a piece in 1961 called Paralytic Child Walking on All Fours from Muybridge, so it's not really appropriation. He actually gave the source. And for Bacon, this image captured the spirit of his own work, in which he considered man to be an animal or a piece of meat, and that in this sense we see this child who is walking on all fours as kind of an atavistic representation of perhaps our days in the past. And I'm going to kind of conclude with a group of 3 artists who have worked under this paradigm of art and science. Mark Dion, who you probably know, New York-based artist, and in 2007 he was commissioned to do an installation at the Natural History Museum in London for the 300th anniversary of Carl Linnaeus, who again was the father of the classification system. Now, when this installation went up, many of the scientists at the museum were outraged that money could be spent on such a kind of frivolous reconstruction of a studio from 300 years ago, or textbooks or pictures, etc. But also as part of Mark Dion's work, he also does field work. His work, although is artistic and he's considered an artist, his work overlaps with strategies in the scientific and archaeological worlds. So one of his field trips had to do with driving as fast as he could down one of the highways in London, and he had attached to the top of his roof, like our speaker about the zebras here, he had flypaper on the top of his roof, and he had nets out the window, butterfly nets, to see if he could capture airborne insects or other life in London. And he brought this information back, took the flypaper off, sent it to the lab, and hence he discovered 2 new species. One a beetle and the one a beetle and the other a wasp. So much for the scientific method, which is another reason why scientists should let artists in their laboratory, because it's like the canary in the mine. They're going to do something that is completely out of the ordinary, something that is unexpected. Another example are Heather Aykroyd and Dan Harvey. and they do what are called chlorophyll photographs. Again, sort of by circumstance, Heather and Dan were sitting in front of their house, and they saw that the ladder, which had been outside and then moved, had sort of changed the color of the grass it was lying upon. And they got the idea that grass, because of its chlorophyll content, could be used as a photosensitive surface, and they began making these large-scale images, which this one is state green grass, clay, and wood. And you can see this one as a biochemical living photograph. And finally, I'd just like to end with a couple of images of my own work from 2009. This was a project I did at Exit Art in New York City when I decided I would grow vegetables in the gallery in these containers that were put together with all kind of prefab parts from the Container Store. And these plants were grown using LED lighting. They were red and blue, kind of creating a fall sun. When the plant was in the box, it looked like it was fuchsia. When I took it out of the box, it was green. And this was based on a little research that I had been doing about what space stations were beginning to do in order to grow food in space. And the fact that LEDs have a low carbon footprint, and if astronauts are going to be out there for a long time, they're going to need some green vegetables. So last year, before the space station was dismantled, a Japanese astronaut was sent up to the station to grow cucumbers, but he was not allowed to eat them. My vegetables, I didn't eat them either because I didn't know whether they would be good for me. But I don't have an anti-gravity tank. If anyone has one and wants to invite me in, I would be happy to perform this experiment. And these are all the images of the piece. None of this is photoshopped. This is what the piece looked like. I would go there every few days and watch the plants grow. Thank you.
Thanks, Suzanne. We've got a few minutes for questions here. See the questioner. No question?
No question.
But I don't get name or whether it's already
One question.
Suzanne, thank you very much for this talk that can shed another light on
How we probably should use or understand the word aesthetics, which is, if I understood you right, not that much just the concept of visibility, because arts are not just related to the eye, as we know. But the way you pointed it, would you agree that you would say that aesthetics is much more a specific way to see, which is then related to the media we choose, to the methodologies we want to see, to the techniques and the theories that are involved in? And if so, how would you then, I mean, the way you explained it, I thought, okay, then another distinction maybe blurs, which is that, for instance, sciences are objective and arts are not, and so on and so on. Assume this is all the case. How would you then describe the difference between the studio and the lab, or the shifts between it?
That's very interesting, since I'm just built a laboratory. And the reason for that is that art uses as its data bank every other discipline. It uses popular culture, it uses Gestalt psychology as a minimalist, it uses geology as in Robert Smithson, it uses anthropology as in Susan Hiller, and it uses the sciences as well. It doesn't use these disciplines to make science, because we're not making science in the science lab, and it's not even called the science lab. It's called the Nature and Technology Lab. And it is about using some of the processes and concerns that living matter addresses now. Since science has come out of the laboratory and into our lives with everything we do, these kinds of questions about the aesthetics of care, about food production, about pollution, about longevity, about pharmaceuticals, about transgenics. These are questions that we are looking at from both the scientific, aesthetic, and social science perspective.
I'm just wondering if you have a sense of the degree to which the potential of artistic and scientific collaboration is based on the aesthetic sensibilities of the artists that go into the lab versus their just ability to produce good ideas. Because some of the examples you use, I'm thinking specifically of using grass as a template for photography and portraiture, and also and driving around with flypaper on your car. Those are just good ideas. And they could have been executed totally ineptly, and they still would have been recognized as good ideas. So to what degree do they need the aesthetic sensibility versus just to be thinking outside the box?
Very good question. I think they are good ideas, and artists have lots of good ideas, but they don't have places where they can enact them. Many laboratories do not want artists coming in because they are unpredictable. I do think that, that the work I also showed had a whole conceptual basis to it. And because art is what happens in an art gallery, as it is contextualized, then there is this idea of aesthetics. But this is my predilection and also what I chose to show. I could have shown you a lot of things that didn't look like anything, okay? And that's where I think art and science experiments fail, is when the art has no body, that it just has an idea.
Thanks. I have one final question for you. I wondered what you meant by, with the flypaper thing, driving the car around. And you said, so much for the scientific method. Are you suggesting that scientists should give up their rigor and their studies, instead just have mad artists running around throwing out weird ideas and jumping off of cliffs?
It's neither nor, okay? It's about invention, which is always something that breaks the rule. And I think that scientists doing good science are invaluable. And they should do what they do. But every once in a while, there is somebody who doesn't do what he's supposed to do, or she doesn't do what she's supposed to do. And I'll give you Alexander Fleming's point here. Or mirror neurons, or the vulcanization of rubber, or many other kinds of chance events. It's through that recognition that something happened that brings together discoveries in art and in science.
Thank you very much.
Yeah.